Hello, welcome to chapter 23, part 1. And in this, I'm going to be looking at the respiratory system. Starting out looking at the general organization. Here we divide up the respiratory system into the upper and the lower. Upper includes the nose, nasal cavity, and the pharynx. At the larynx, the lower respiratory tract begins. And then below that is the trachea, bronchi, bronchioles, alveolar ducts, and alveoli. Now, another breakdown in the organization of the respiratory system is functional organization. We've got what we call the conducting zone and the respiratory zone. Now, of the structures I just mentioned in the upper and lower, they are all conducting zones. In other words, all they do is transport the air. They transport the gas, the air, until it gets to alveolar ducts and alveoli. All right, maybe some respiratory bronchioles. But until it gets down to the last structures, they are all conducting zones. And then the respiratory zones, again, respiratory bronchioles and alveoli. All right, that's where the actual exchange of gases take place. This is where the capillaries surround the tissues and the actual exchange takes place. So that's the respiratory zone. But a great majority of it is the conducting zone. It's just bringing air in. And not just bringing it in, it also warms the air and cleans the air and humidifies the air. It cannot be dry air when it gets down to the lungs. Cannot let the surface of the lungs get dried out. So again, structural organization, upper respiratory to the pharynx, lower respiratory to the larynx down to the alveoli, the respiratory structure of the lungs. Functional conducting zones, okay, and then respiratory zones, the last parts, as you can see here. These bubbles here are alveoli, where gas exchange takes place. Now, what happens to the epithelium as it goes from the beginning to the end of the respiratory system? Well, in the beginning, remember I said one of the things was that it cleaned the air. This is one of the first things to be done. So we have goblet cells, pseudostratified ciliated columnar epithelium in the cat. Sorry. Um, in the upper parts of the respiratory system because it's cleaning the air. It's got a lot of mucus. It wants to grab all the particles off of it. And then as you go further on down, it gets a little bit thinner, so simple ciliated columnar. Again, you're going to get a lot of mucus on those as well. A little bit of mucus left as you go further down. But then as you get to the last structures where that gas exchange is taking place, you're going to have a simple squamous epithelium, of course, because gases have to exchange through those cells so that they have to be very, very thin in order for those gases to exchange, to travel through the cells. So looking at the beginning here, the nasal cavity, we've got the nostril or the nares. The concha, you might remember looking at them, superior, middle, and inferior nasal concha. These were bone structures, if you remember looking at the skull. Quinae at the back. This is kind of the beginning, the start of the nasopharynx at the back here. And then you've got the oropharynx, which is behind the oral cavity, and then down below you have the laryngopharynx behind the larynx. There are, it's composed of frontal bone, nasal bone, ethmoid bone, sphenoid bone, the upper roof of the nasal cavity. Remember all those structures and remember the olfactory region and the bulbs and all that up there. Okay. So looking at the nose and nasal cavity, you've got nasolacrimal ducts up there as well. These drain the lacrimal secretions from the eye surfaces So remember that across the eyeballs. Remember the lacrimal ducts put tears across the eyeballs, and they collect in the corner of the eye right there, and then they drip down into the nasal cavity as well. Um, so like I say, it conditions the air, warms it, cleanses it, humidifies it. Mucus traps dust, microbes, other things out of it. Um, and moist environment humidifies the air. Remember the Pyrenees sinuses as well. Um, they're covered with pseudostratified, or they're coated, not covered, but in the inside. Um, they produce mucus as well, and then they sweep all of the what they collect into the pharynx as well. So from the nasal cavity, we get to the pharynx, and I've emphasized the Y and X because people have a tendency to call it the pharynx. Okay, um, often kind of joked that if you're gonna have that on a question on the quiz, pharynx, and if you say the word pharynx, you're gonna get a C. Anyway, but just remember it's Y and X, not NYX. Okay, pharynx, not pharynx. Um, and basically, this is the throat. Three sections here: nasopharynx, oropharynx, laryngopharynx. The oro and the laryngeal are also shared with the digestive system. Um, so nasopharynx again behind the nasal cavity, not for food, for air. Connects also to the eustachian auditory tube. Um, Here you find some of the tonsils, the pharyngeal tonsils. All right. In the oropharynx, we find the other tonsils, the palatine and the lingual tonsils. And the laryngopharynx, no tonsils posterior to the larynx. Same thing here, the larynx. So past that, we get to the larynx. Again, those structures are shared with the respiratory system and the digestive system. So it goes to the esophagus if it's the digestive system. And then, of course, it's food and water. But the air goes down into the larynx. The larynx is also the voice box between the laryngopharynx and the trachea. Functions of that is for an air passageway um, to help prevent ingested materials from entering the respiratory tract. Um, don't want that stuff going down in the respiratory tract. How that works is with an epiglottis covering the top of the opening of the larynx. Larynx is also responsible for producing sound for speech. Has vocal cords that vibrate during our expiration. The larynx is made of 9 pieces of cartilage, 3 pairs, and 3 individual. All right, so there's 3 pairs of cartilage, which is, of course, 6 pieces, and then 3 individual pieces for 9 total. The ones that are individual are the epiglottis. You can see that flaps down over the covering, okay, over the airway, over the space. It flaps over the space and causes the food to push down behind the larynx and down into the esophagus to the stomach. There's the thyroid cartilage, and this kind of encompasses on 3 sides, the anterior and the 2 lateral sides. This is where we get the Adam's apple, the laryngeal prominence right there. And then there's also the cricoid cartilage. Right? That's way down at the bottom here, and it's kind of like almost like the first ring of the trachea. It's not that, but it's almost kind of like that. Doesn't really have much other functions. Cuneiform, cuniculate, and arytenoid cartilage are the ones that are all paired. So there's one on the right and one on the left side. And these are the ones that kind of come together. They're much smaller than the other ones. They're attached to ligaments and muscles so that they can open and close. They pull open and they pull closed. The airway so that the air, when it's being pushed out, is directed over the vocal cords. Okay, so they kind of close off the airway, except for the slit where the vocal cords are, and this makes the air have to push over those vocal cords, with greater force and vibrates them. Okay, so the airway really, when we speak, is much tinier than when we breathe, and this is why we can't breathe in while we breathe out. So we're pushing the air out, we're controlling the air, the pressure of the air outward um, when we speak. If we try to speak while we're bringing air in, doesn't really work that way. You do make sounds, obviously, but it's definitely not the same kind of thing. You can see here how these smaller cartilages, the muscles and the ligaments that hold them all together, kind of pull them shut, close them shut. Okay, and on either side, so you got the vocal cords here, which are ligaments, elastic ligaments, and you got the vestibule on either side, which are like flaps, like wings or something that kind of close off all of this area on either side of the vocal cords. So it kind of comes down to a little slit between those two flaps where the vocal cords sit, and so the air has to be pushed through those. And so when it gets pushed through those, the vocal folds or the vocal cords right there. These flaps here are the vestibular folds. Okay, so here it is with all of the tissues on it, the soft tissues on it, and the ligaments, muscles, and such, epithelial. Okay, so let's try. There's a video that I've got here. Let's see if we can get that opened. Fortunately, I don't think I found a way to. So they shove this little tube down someone's nose. Okay, so again, this is all humidifying the air and cleaning the air as it goes through. Um, Station tube open. So you've got the soft palate. So you're going through the larynxes, and the person speaking. So the person speaking when it closes off like that. So you can see, bring it down, keeps going down through the pharynx, and there's the epiglottis. So here you can see the voice box right there. Okay. You can see when somebody speaks, while squeezed together. Okay. And if someone brings their voice up, she's bringing her voice up, it stretches it, right? So here you're looking at arytenoids, moves them together. Okay, so you can kind of see as she's bringing her voice down, it goes slower. I mean, they go shorter. So this is using a strobe light so you can see them moving. You can actually see them moving slowly. Okay. Vocal cords change as she changes her pitch. So she's going up in pitch as she goes up in pitch. So when we breathe in, the vocal cords, the muscles relax, opens up the folds, opens up the vocal cords, and you can see here that you close it off to make sure that the air is going over the vocal cords when we speak. Okay, let's get back to what we were doing. So from the larynx, we go to the trachea or the windpipe, open tube connecting the larynx to the main bronchi. So this is the one long stretch of the respiratory tract that is opened with the C-shaped, this is a C-shaped here, ring of cartilage, and they're held together with these bands of connective tissue. Okay, looking a little bit closer on this. So they're held together with bands of connective tissue in these C-shaped um, cartilage rings. They make sure that it stays open all the time. So we want it to be open all the time, so we don't have to keep with every breath. We otherwise would be opening, closing it constantly. We don't want that to. We don't want to have to be doing that. So as you can see here, we're talking about the trachea, and the trachea goes down further into the thoracic um, cavity, and then it splits. As soon as it splits, it's not the trachea anymore. Now we're talking about the bronchi. Okay, bronchial tubes, the bronchi. That first split gives us two bronchi, one to the right, one to the left. One's going to the lung on the right, one's going to the lung on the left. And so they are the main bronchi. The first one's primary main bronchi. Okay. Looking at the histology of the trachea wall, so you can see here there's a lot of mucus in there to capture a lot of particles and potential and microbes and whatnot. And then it gets swept up. You can see the cilia on the top of the pseudostratified ciliated columnar cells. Push that mucus up and the stuff on it up through the larynx and then down into the esophagus where they go into the digestive system. Okay. So we've got the mucus layer on top, then this epithelial layer, submucosa right there. So there's a little layer behind that pseudostratified columnar called the lamina propria, and then the submucosa with blood vessels, nerves, and whatnot come in. And then if we keep going further on out from there or further, I suppose we're looking at more superficial. Okay, so the mucus would be deeper and the inside of the trachea would be deep. So we're going to superficial here. Cartilage on the trachea and then elastic tissue connecting the cartilages. Okay. So what happens then is we got the primary bronchi split to the right and left lungs. All right. It narrows. You can see here they get narrower than the trachea. Then the primary bronchi break off into secondary bronchi. Okay, these are the secondary bronchi. And each of these is going to a different lobe of the lung. So the left one has got, the left side has got 3 lobes, okay, and the right side has got, or the right side has got 3 lobes and the left side has got 2 lobes. All right, the left side is 2 lobes and the right side is 3. And those are the secondary bronchi. And then it splits again and called the tertiary bronchi. These are regions of the lobes, okay? And then they continue to branch and branch and branch even more. We lose the cartilage because it gets smaller. It gets to the point where we don't need the cartilage to hold it open all the time anymore. It gets small enough we don't need that. And then we get to the final, or the terminal bronchi. So the last bronchi with cartilage are the terminal bronchi. Okay. The bronchioles come after the terminal bronchi. Okay, bronchioles are without cartilage, and then they keep branching. You can see bronchioles here, right? And they're wrapped in smooth muscle. The last of the bronchioles are the respiratory bronchioles. So the respiratory bronchioles then subdivide to the alveolar ducts. And alveolar ducts are these last ones that lead to these big bubbles here. I'm calling them bubbles. They look like a bunch of bubbles. And these are the alveoli. And it's on these alveoli that the capillaries surround them and the gas exchange take place. All right. And again, the epithelium, by the time we get down to those, in the bronchioles, respiratory bronchioles, you're looking at simple cuboidal. By the time we get to the alveoli, where the gas exchange takes place, we're talking about simple squamous. Okay. So looking a little bit more closely at these alveoli, you can see that there's different types of cells in here. There's type 1 cell, type 2 cells. and there are macrophages, okay? Type 1 cells, type 2 cells. Type 1 cells go to make up the lining, okay? Type 2 make up the surfactant. There's a special thing called surfactant, and it's a special substance that keeps the surface tension low. Now, surface tension, for example, in water, remember, there's all this stuff is sort of in a water solution of one kind or another. And remember, water, right, has a high surface tension. It's hard to break into water. You got a drop of water, it's hard to break into it. This is what causes water to bead up. You see water bead up, especially when it's on a waxed car. Wax is a hydrophobic substance, so it gets water on it, it's going to repel it. Okay, it's going to repel it, and in doing so, it makes it kind of fold up on itself and bead, as we say. And this is surface tension, so we have to have that lower because we don't want that interrupting the structure of the alveoli. It's very fine, very delicate in there. And so we have the surfactant. And you can see here it's the blood vessels, and there's a single erythrocyte going through the blood vessels, which, the capillaries. And they go through one at a time, kind of not piled on each other, because we need the surface area of each one of those erythrocytes to do the gas exchange properly. Here's a very close up of it. So in here, out here is the outside of the inner layer, as it were, of the alveolus. Okay, the lumen of the alveolus, and it's basically still outside the body, right? When you think about it, it's still outside of the body. And then the epithelial layer, okay, and then we got the epithelial layer of the capillary, and then we've got the erythrocyte that has the hemoglobin in it. So the oxygen has to get through all of that, and the carbon dioxide has to come out of all that for this gas exchange to take place. Now, in order for this to happen, it's just a simple diffusion. There aren't any proteins that are grabbing these molecules and shoving them either side. It's actually diffusion, just a simple chemical diffusion. And so these things take a long time. If we want it to be done efficiently, we've got to spread out all the blood going through there so that it goes through erythrocytes one by one through the capillaries, okay? We've got to spread them out. And there has to be lots and lots of surface area for them to take their time going through there. And that's what these bubbles provide. They provide a lot of surface area so that it can happen at a rate that's most efficient. Doesn't have to be necessarily fast. Like I say, it's not fast, but it has to be very efficient so that the blood gets the maximum amount of oxygen that it can and drop off the maximum amount of carbon dioxide that it can, right? Looking at the respiratory zone, we have the respiratory bronchioles, alveolar ducts, the alveoli. Again, looking at those type cells, type 1 cells, the squamous cells make up the surface area, and that's what most of the surface area is made up of those cells. Type 2 cells make up the surfactant, like I said. And the other thing in there are macrophages, and you can see them teeming around in there. They're devoted to cleaning up anything that happens to make it all the way down to that level. Hopefully it's all been caught by the mucus on its way down, but things still get all the way down there. Looking at the lungs in general, okay, they kind of have this cone shape to them. There's the apex. Here we're dealing with the right notation. Remember, the heart, the apex was on the bottom and the base was on the top. But so the apex in a peak like that, and also the most superior part of the lungs, and then the base in a wider area down there at the most inferior part of the lungs. So the right lung here has got, this is the anterior view. The right lung here has got 3 lobes. The left one has 2 lobes. And it's got what they call the cardiac notch on the left. So the left, remember when we looked at the heart is a little bit, it's right in the center for the most part, but it goes a little bit to the left. Okay, so the left lung is a little bit smaller and only 2 lobes. It has to make that space for the heart. You can see the cardiac notch, cardiac compression. So it also has a hilum to it. Oops, what's going on? Um, and there you can see that right there on the back where the blood vessels go in, and also where the bronchi, you know, enter the lung tissue right there. Okay. Bring that up again. Okay, next slide. So when we get to circulation, we look at circulation of the lungs. There's actually two blood supplies to the lungs. One is to provide what the lung is there for, so the blood that's coming to it is to provide what the function of the lungs are for. Remember, function of the lungs is to replenish the oxygen and eliminate the CO2, or lower the CO2 and add the oxygen. Okay, reoxygenate the blood and decarboxylate the blood. Um, and so that's called the pulmonary circulation. So these are the ones that go through those capillaries that we looked at. But the tissues of the lung, remember those, all the tissues that we looked at, the uh, muscles and the epithelium and all of the bronchi, of the trachea and all, they need to have their own blood supply too. And so that's the blood supply is bronchial circulation. It transports that oxygenated blood to those tissues. All right. So here we're looking at more of a close-up of the pulmonary circulation of the lungs. Um, there we go. And how the blood comes in. Remember, so it comes in on the arteries that are deoxygenated. We're talking the pulmonary circulation now, remember. Comes in on the arteries with deoxygenated blood, goes through the capillaries, picks up oxygen, in reverse of what we see in the systemic side, and comes back through the veins that are oxygenated, right? Um, so looking at this too, there's a lymph drainage. They are located, lymph vessels and nodes are located within the connective tissue around the bronchi. And the pleura, the cavity surrounding the lungs also. Again, getting rid of excess fluid is important for the lungs. Fluid buildup in the lungs is a bad thing. It can, inside the lungs, it can block the transfer, the exchange of gases inside, and on the outside, the fluid can hinder the expansion and the movement of air in and out of the lungs. Okay. Um, just kind of finishing up a little bit here, we talk about the membranes in the pleural cavity. Remember, this goes all the way back to chapter 1. We talk about serous membranes, remember? So there's the visceral pleura on the outer surface of the lungs, and the parietal pleura that lines the lungs. Again, parietal and visceral sides of the serous membranes, right? The pleural cavity itself, located between the visceral and parietal serous membranes. This is a space in between the two. So we're looking at where the fluid is produced, and lubrication occurs. Remember, serous membranes were all about lubrication. So we're talking about pleural pressure. And we'll talk a little bit about that. There's a pressure outside the lungs that help them to expand. All right. And the serous fluid here, not only is it for the lubrication, but it helps those two surfaces stick together. So the expansion pulls on the lungs and opens them up. And you can kind of see that here with that intrapleural pressure. This is something we talk about a little bit more in the second part. But lungs remain inflated because of that pressure. It always has to be a little bit less on the outside of the lungs because the lungs are very springy, very spongy. And after they expand, they're going to just spring back from where they began. Okay, they're just going to spring back. And um, so without a constant negative pressure to keep pulling them open, even after you exhale, they would collapse. Okay. Um, And so that's how they remain inflated all the time. And that's what I've got for now. And from here, I am going to go into part 2 and talk a lot more about the functioning and gas exchange and such. But this is the end of part 1, and see you later.